Welcome to lecture number 4. This is part 2 of the microbial lifestyles and behaviors lectures. And what these lectures are meant to do, and really what unit 1 is, the whole unit is meant to do, is to get you to see the world more like bacteria see it. You can't think about senses like vision and hearing. You can't think about brains. You have to think about chemistry. And we have to think about how they solve problems using chemistry. And so some of what we saw in lecture 3 was things like how do they get energy? What sorts of problems do environmental conditions give them? Like, is the water too salty? They can't get water. Is it too hot? Their membranes get too fluid and their proteins denature, stuff like that. We saw the idea that they have to be pretty specialized about what they use as their carbon sources and their source of electrons and their source of energy. And different bacteria will do that differently. And so now, in this lecture, we're going to continue that kind of idea, and we're going to look at more of the energy metabolism. We're going to kind of finish that, and that goes to alternate electron acceptors and fermentation. And you will have, of course, seen fermentation before when you've looked at things like muscle cells, but bacteria have a lot more diversity to the fermentation they do. And then we're going to go to biofilm and quorum sensing. This whole lecture is going to have more detailed examples than you might be accustomed to. And what I want you to do is work with the examples, be able to explain the examples. I'm not going to ask you to remember all of them. I don't care if you remember most of them. What I care about is that you can tell me what they are for and what they mean and what they're telling you. Because the purpose of working through all these examples is to get you to fully understand what I'm trying to show you. So we're going to go through examples for the purpose of understanding what bacteria are going through. So this is the list of topics from lecture 3. We've gone through water, temperature, oxygen, pH, nutrients, and now we're at energy metabolism and electron acceptor. So there are two broad ways bacteria can generate energy. One is substrate level phosphorylation, and that's where they rearrange molecules with phosphate groups on them until they have a high energy bond, that's what we used to call them, that they can use to phosphorylate adenosine diphosphate, or ADP. And then the product of that reaction is ATP, adenosine triphosphate. And that is the source of energy that a lot of enzymes are able to use. So substrate level phosphorylation is what happens in glycolysis. And that's a way of making ATP, among other things. What you've also seen is respiration, where we oxidize glucose or we oxidize lipids, and that gives us reduced electron carriers like NADH and FADH, and those go to the electron transport chain, just like what we saw in lecture 3. And as they go through the electron transport chain to an electron acceptor, such as oxygen, that flow through the electron transport chain is a source of energy, and we harness that using the complexes in the membrane to pump protons out of the cell. And that energizes the membrane, that puts a strong proton motive force on the membrane, and that's something we can harness in our mitochondria to make ATP, and that is all based on the bacterial systems that use that to make ATP. So when you think about respiration, that's oxidative phosphorylation. It's oxidizing, taking electrons off of our electron donors and using the flow of electrons to phosphorylate ATP. And it is a complicated process, and it's not something you can explain in 30 seconds. It does have a lot of parts. So This would be substrate level phosphorylation, where you start with molecule A, change it, phosphorylate it, and then we can, what happens in glycolysis, for example, what happens in glycolysis is we rearrange the molecule until we have high energy phosphate bonds, and then they react with ADP to make ATP and the final product. And so if this were glycolysis, this would be glucose, and this would be pyruvate. This would be phosphoenol pyruvate. And there's a lot more going on. And then oxidative phosphorylation, again, is energizing the membrane by pumping protons out. And what that does is that builds up positive charges on the outside. And so the protons kind of stick there, and they're trying very hard to get back in because they're attracted to the negative charges on the inside. And then we can use ATP synthase complexes in the membrane to make ATP. And when we do that, we kind of lose that energy. So we go from a high proton motive force here, and we spend some of it to make this ATP. So we have not as many protons outside the cell. So that's oxidative phosphorylation. Generally, oxidative phosphorylation is more efficient and generates a lot more ATP than than substrate level phosphorylation does. And so what you would have seen in human physiology was a comparison of respiration using oxygen versus fermentation, and how much ATP you can get from one glucose molecule, and it's vastly different. And so the difference between these is huge, and we're really going to need to work through this a little bit in this lecture. So respiration, we can define respiration as the process that uses an electron transport chain and an external electron donor, so in our case glucose, an external electron acceptor, that would be oxygen, and a proton-dependent ATP synthase. And all of those things working together make ATP. And so what you would see in human physiology is a very specific type of respiration called aerobic respiration, and that's when we use oxygen as the electron acceptor at the end. But bacteria and archaea have a lot of different versions that use different electron acceptors other than oxygen. So that's part of what we'll see today. And then fermentation is a completely different system where ATP is made only through substrate level phosphorylation, and we'll work through some of that as well. So this again is part of what we saw last time. Electron transport chain with the complexes that pump protons and the ATP synthase complex. So I want to walk through basic glucose metabolism as it would happen in a mitochondria or a gamma proteobacteria, or alpha proteobacteria, which is what mitochondria are related to. So this should be familiar to you. This is glycolysis. And the way I like to write these pathways is I list what goes into the pathway, so what is necessary, the molecules that have to be there for this to happen, and then the molecules that are created through glycolysis. So these are all the inputs to glycolysis. These are all the outputs from glycolysis. I color code electron carriers, so NAD, NADH. These are two different versions or two different forms of the same molecule. Here, well, this is an electron carrying molecule. This is the form when it's carrying 2 electrons. This is the form when it is not carrying those 2 electrons. And then blue is what I use for the carbons, so we can keep track of the carbon from glucose. All the carbon from glucose ends up in 2 molecules of pyruvate at the end of glycolysis. And so I'm going to use this same color scheme to go through the steps after glycolysis. the first thing that would happen would be pyruvate oxidation, and everything here would be multiplied by 2. So there'd be 2 NAD+, 2 NADH, 2 pyruvate, 2 acetyl-CoA, 2 coenzyme, and 2 carbon dioxide. And that comes from the fact that there are 2 pyruvates that result from glycolysis. Remember, glucose is a 6-carbon molecule, pyruvate is a 3-carbon molecule. And so there would be 2 of them, so all of these would be multiplied by 2. And again, we get 2 NADs that pick up electrons from pyruvate, so that at the end we have acetyl-CoA and NADH. So now this is carrying 2 electrons taken from pyruvate. One of the carbons from pyruvate goes to carbon dioxide. That's as oxidized as carbon can be. So if you see carbon dioxide, you know that the electrons have been taken off of that carbon, as many as can be taken off. So it is that carbon has been oxidized. That's what it looks like. And then acetyl-CoA are the other 2 carbons attached to a coenzyme. So that's what comes out of pyruvate oxidation, and then that acetyl-CoA goes to the citric acid cycle. And in our own cells, this happens in the cytoplasm, and these happen in the mitochondria. And so the citric acid cycle is more complex, but it starts with acetyl-CoA and all of these oxidized cofactors that are electron carriers that aren't carrying electrons, and then up here, electron carriers that have gained electrons. And so what this process accomplishes is taking the 2 carbons from acetyl-CoA and turning them into carbon dioxide. And again, that's as oxidized as carbon can be. That means we've taken the electrons off of that carbon and put them onto the NAD and FAD, resulting in NADH and FADH2. These are the electron carriers in the form when they are carrying the electrons. Okay, so let's go to the next slide. Again, the idea is that the electron transport chain is going to take the NADH that was made in glycolysis and pyruvate oxidation and the citric acid cycle and some of the FADH, and those go into the electron transport chain. So we write this overall reaction as glucose plus oxygen, and that's really what it is, but the only part that goes into the electron transport chain are the electrons from NADH. So NADH would go here and donate electrons. So I put this here in this kind of conceptual electron transport chain, and they pass the electrons to the quinone, which passes the electrons to cytochrome, which pass the electrons to oxygen. So this is the central energy metabolism in any organism that's using aerobic respiration. They're going to have something similar to this with glycolysis, some kind of pyruvate oxidation, and some kind of citric acid cycle. Bacteria might use something very different from glucose to grow, and so they might need to, they might have modified systems, but this is the general idea. And so what you should know is these colored molecules. So the NAD in both of its forms and all the carbon containing molecules. So this, these 3, and these 3. From our point of view today, those are the molecules that matter. And you should know which ones are products and which ones are reactants. What is an input and what is an output. And so the last thing for this video is we're going to look at the type of organism that would use these systems. And those would be obligate aerobes, facultative anaerobes, and microaerophiles. These are all going to use some kind of aerobic respiration, analogous to what happens in our own mitochondria. And that's where we're going to leave it for this video. Again, as we go through all of our examples, we are really trying to see the bigger picture, and we're trying to see what the examples illustrate. So I want you to work towards understanding. Make sure you understand every slide in this lecture. Okay, see you in the next video.